Welcome to The Explainer. Today, we're diving deep into the most complex and, honestly, the most elegant code ever written, the code of life itself. We're going to see how we learn to hack it. Let's get into it.
Just think about this for a second. What if you could open up the instruction manual for a plant, or an animal, or even us, and not just read it, but edit it? What if you could find a typo that causes a disease and just fix it? Or maybe add a brand new feature, like giving a plant the power to make its own vitamins. This isn't some sci-fi movie plot. This is the reality of genetic engineering.
So to understand how to edit this code, we first have to get a handle on how life's operating system actually works.
Okay, I want you to think of every living cell as this tiny, incredibly sophisticated computer. And just like your phone or your laptop, it runs on software. This biological software, it's DNA, and it holds all the instructions for everything the cell needs to do to build, maintain, and copy an entire organism. Now, the way information flows in this system is so fundamental to biology, it's actually called the central dogma. It works like this. First, you've got the DNA. This is the master blueprint, right? It's locked away super securely inside the cell's nucleus for protection. But when the cell needs to get something done, it's not going to risk damaging the master copy. No way. Instead, it makes a temporary, disposable copy of a specific instruction. That copy is called RNA. This RNA, the working copy, then leaves the nucleus and goes out to a cellular factory. There, its instructions are read, and they're used to build a specific protein. And proteins, well, they're the action heroes. They're the little machines, the enzymes, the building blocks that do all the actual work in the cell. So DNA to RNA to protein. That's the core process. That's the whole game.
So when we talk about editing the code of life, what we're really talking about is editing genes. A gene is just one specific section of that massive DNA blueprint. It's the single instruction for building one of those action molecules, one specific protein.
For the longest time, this incredibly elegant system was just a total black box. Before we could even dream of writing the code, we first had to figure out how to read it. And that was a truly monumental task.
Just to give you a sense of the scale here, the human genome, our complete set of DNA, is made up of about 3 billion DNA letters. I mean, try to imagine reading a whole library full of books written in an alphabet with only 4 letters, and with absolutely no spaces or punctuation. That was the staggering challenge scientists were up against.
So how on earth do they do it? Well, the big breakthrough was this brilliant method called Sanger sequencing. Conceptually, it's actually pretty clever. You start by making millions and millions of copies of a DNA strand. But here's the trick. Each copy is designed to stop at a different letter, and that final letter gets tagged with a unique color. Next, you sort all these fragments by size, from the shortest to the longest. And finally, a laser zaps each fragment as it goes by, reading the color of that final tagged letter. So if the shortest fragment ends in a green A, and the next one's a red T, and the next is a blue G, boom, you've just read the sequence, ATG. You just have to scale that up, and you can read an entire genome.
Reading the code was absolutely revolutionary. But the next leap, well, that was even bigger. Learning how to actually edit it.
This is the huge difference between being a passive reader and becoming an active editor. Sequencing let us read the book of life. Genetic engineering gives us the pen to rewrite it, to fix the typos, to add new sentences, or even change entire chapters.
To edit DNA, you need a basic toolkit. First up, you need what are essentially molecular scissors. These are special proteins called restriction enzymes, and they are unbelievably precise. They recognize very specific sequences of DNA letters and cut the DNA only at those spots.
Okay, so you've got your scissors. Next, you need some molecular glue, and that's an enzyme called DNA ligase. Its job is to repair any breaks in the DNA, which means it can effectively paste different pieces of DNA together.
And finally, you need a delivery truck. For this, scientists often borrow a really handy tool from bacteria called a plasmid. It's just a small circular piece of DNA. They can use their molecular scissors and glue to load a new gene onto this plasmid, and that plasmid then acts like a vector, a delivery truck, to carry that new genetic code into a target cell.
So, with this incredible toolkit in hand, what can we actually build? Well, the applications are already all around us. And a huge one is in agriculture. One of the most widespread uses for this tech has been in farming. And this isn't some new thing. I mean, just look at this data from all the way back in 2012. Even then, you can see that a huge chunk of major crops like soybeans and corn were already genetically modified, usually to make them resistant to pests or weeds. And that trend has just kept going.
Yeah, this capability has been around for a while. Scientists first managed to stick a foreign gene into a plant way back in the 1980s. And as this quote from a textbook points out, since then, we've figured out how to do it in more than 120 different species. That just shows you how mature this field has become.
So let's look at a classic example to see how these tools really work. Golden rice. The problem was simple but devastating. Millions of people suffer from vitamin A deficiency. And rice, a food staple for so much of the world, doesn't make beta-carotene, which is what our bodies turn into vitamin A. Scientists looked at this and saw it as a broken assembly line. The rice had all the starting parts, but it was missing a couple of key steps, a couple of key enzymes. So what did they do? They took out their toolkit. They borrowed the gene for one missing enzyme from a daffodil and another from a bacterium. They used their delivery trucks, their plasmids, to insert these new genes into the rice genome. And just like that, they fixed the assembly line. The result? Rice that can now produce its own beta-carotene, giving it that famous golden color.
Our ability to read and write the code of life hasn't just given us new tools. It has literally opened up entire new fields of science. So, what's in the next chapter of this story?
Well, it's fair to say we've gone from editing a single word to trying to understand the entire library. Our ability to work with genes gave us genomics. But that was just the beginning. That led to proteomics, the study of all the proteins, and metabolomics, the study of all the chemical byproducts of a cell. And the grand finale, so far, is systems biology, which tries to mash all that data together to understand the organism as one big, dynamic system.
So here we are. We've gone from being simple readers of the genetic code to being its authors. We now have an incredibly powerful toolkit that lets us rewrite the very instructions for life itself. And as we turn the page to this next chapter, the big questions aren't just scientific anymore. They're about us. They're about society. With this incredible power, what kind of future are we going to choose to write?